Welcome to today's Matchmaking webinar for the ARPA-H Breathe program, which stands for Building Resilient Environments for Air and Total Health. My name is Chris Smith, and I am a Systems Engineering and Technical Advisor, contractor to ARPA-H. The purpose of today's webinar is to provide organizations and entities with an opportunity to exchange information about their areas of expertise and their teaming needs for the Breathe program. We will begin today's presentations with a brief introduction from Dr. Jessica Green, the Program Manager for Breathe. We will then transition to lightning talks. Finally, we'll close with key next steps. And with that, I will turn the webinar over to Jessica for an introduction to ARPA-H and Breathe.
Hi. So briefly, the mission at ARPA-H is to accelerate better outcomes for everyone. I want to encourage everybody today to review the Proposers Day webinar materials because we went over in great detail everything about the Breathe program. And I also wanted to encourage everyone to read through the FAQs. We're getting a lot of questions about the Breathe program, and we're answering those questions in tranches. So we're very closely completed with questions that have come in to date, but we'll keep answering questions. And then last thing that I wanted to share is that before we dive into the lightning talks, I wanted to emphasize a key aspect of the program, and this relates to a question that keeps coming up. So we are receiving inbounds with people asking if for this program we'll be funding the advancement of sensor technologies for monitoring particulate matter and chemical pollutants in the indoor environment. And this is out of the scope of the Breathe program. That being said, we are not discouraging the use of bringing together different types of indoor air quality solutions. So if there are existing air quality sensors that measure for air chemistry or particulate matter, we are encouraging teams to bring those to the table and to couple that with a newly invented air biosensor. And we also are open to the idea that there may be some types of sensors that can be used in the indoor environment to assess human health and well-being, and look forward to seeing what people bring to the table with their abstracts. And without further ado, I'm going to turn it back over to Chris.
Thank you, Jessica. We will now begin the lightning talks. If you are a presenter, please unmute your microphone when we call your name. We will share your single slide submission on the webinar and invite you to provide a 3-minute presentation. We will verbally let you know when 1 minute remains. I would first like to invite Peggy Lai from Harvard Medical School and Massachusetts General Hospital to give her talk.
Right. Hello. Good afternoon, morning. I represent a research group based at Harvard Medical School, Harvard School of Public Health and affiliated hospitals. Our research group has over a decade of research on indoor air quality in schools to improve health outcomes, with recent clinical trials published in high-impact journals such as JAMA. I think what's unique about our research group is that we have experience in clinical trials of environmental interventions, such as portable HEPA cleaners in schools. In recent years, we've published on interventions focused on indoor allergens and asthma. And more recently, because of the COVID-19 pandemic, we've developed multiple approaches to rigorously measure viral exposures in schools. And so as one example here, I present some preliminary findings from one of our studies where on the x-axis, you see every column is one classroom. On the y-axis is every virus detected in a single air sample. And so you can see that in some classrooms, we have as many as 11 different viruses detected over the period of 1 week in one classroom. And so what I think we offer as a partner in this endeavor is we are highly experienced in clinical trials focused on environmental interventions in schools, as well as concomitant assessment of health effects. We are able to sample buildings while they're in use. Many samplers are often extremely noisy, and so this is a problem, for example, in classrooms where children need to learn. And also, we have developed very vigorous viral and other pathogen detection methods for indoor aerosol aerosol monitoring. So I'm looking forward to finding partners to improve indoor air quality in schools. Thank you very much.
Great, thank you very much. Next up, we will invite Brian Gilligan from General Services Administration, or GSA.
Great, thank you very much. As mentioned, my name is Brian Gilligan. I'm Acting Director of the Office of Federal High Performance Green Buildings at the U.S. General Services Administration. GSA is a federal agency, and as such, we can be a TA3 partner on any and all proposals. GSA acts as the government's landlord. We're the largest single portfolio of office space in the United States. We manage over 360 million rentable square feet of space. A little over half of that is in 1,600 plus federally owned assets or federal buildings. The rest is in about 6,500 leased assets. Our portfolio is spread all over the US in over 2,000 communities and in every single state, territory, and climate zone of the US. In total, we house about a little over 1 million federal employees from all different civilian agencies. And our portfolio ranges from highly sophisticated to less advanced facilities that require some modernization. But if you look on the sophisticated side, our Smart Buildings program incorporates 168 facilities into a platform called GSA Link. It's about 50% of our own floor space. And that's an integrated national control platform for all of the local building automation systems in those 168 buildings. It allows automated fault detection across a vast network of control points and trending of vast quantities of system performance data. It's been called the national treasure by Carnegie Mellon University because there's really nothing else like it in the US. Our own facilities represent a mix of uses, as you can see on the right. The majority of the space, about 90%, are offices and courthouses. The average age of those buildings is around 65 years, but we have a mix of very historic and very modern facilities in that. In those offices and courthouses, we host typical office workers, public assembly, legal and law enforcement activities, secure communications, and many other uses. We also have other specialized buildings, including labs and land ports of entry. The ports in particular manage most of the overland traffic between the United States and Mexico and Canada. We do also have 2 standalone childcare facilities, which you'll see on that table.
1 minute left.
But there's about an additional 800,000 square feet of childcare around our federal office buildings. Finally, GSA has a well-established research program, as you can find from the links that we've provided. Our Center for Emerging Building Technologies manages what we call the Green Proving Ground, where we partner with national laboratories to deploy and evaluate commercially viable technologies and see if they're ready for prime time in our buildings. The office I work in, the Office of Federal High Performance Green Buildings, sponsors research on the influence of indoor environment on human health, and we partner with labs and other academic institutions like Carnegie Mellon, University of Arizona, Rensselaer Polytechnic to conduct human subjects research in our buildings. We're also in the process of launching a Health and Buildings Roundtable, where we hope to leverage similar work across the public and private sectors. Again, we can work on any proposal and are happy to entertain suggestions. Please feel free to reach out for me. Thank you.
Thank you very much. Next up, we have Rick Rinzonsky from Exemer.
Hi, everybody. My name is Rick Brzezinski, and I am the co-founder and CEO of Exemer. Exemer is a dual-use biosecurity company that uses proprietary UVC technology to prevent the transmission of infectious disease in the air and on surfaces. We are potentially a TA2 and 3 partner. So since its inception, Exemer was formed about 36 months ago. We've been awarded 6 government R&D contracts relevant to the breathe topic from the DOE, EPA, and DOD. We've also formalized contractual partnerships with 8 leading commercial organizations and 4 academic research institutions, including Columbia, NC State, Purdue, and UC Boulder. We've been blessed to assemble a world-class multidisciplinary science and business team, and we've got skills in everything that you see on the slide here. I'm not going to go through each one in particular and take up all the time, but near-field infection protection is the basis of Exemer's technology. This creates a biosafety barrier against aerosol transmission in close contact environments. We look at this as a source control solution that follows people around the room. Our theory and our thesis is that no matter how clean a room is, it's only as clean as the last person who comes in and sneezes in the room. So just keep that in mind. It's a layered protection strategy that works together with built environments, and it enables us to deploy stationary and portable and mobile devices within an environment that layers in and communicates with the built systems. So we also have CFD and modeling simulation capability that is used to design all of our systems. And through our partnership with Purdue and its air quality chamber, we do IAQ risk assessments, and one of our chief scientists, Dr. Blatchley, has written and published a lot of information on these risk assessment capabilities. We are also partnered with Zeteotech, who is a leader in biological mass spectrometry sensors, and with our capabilities in IoT and software applications, we believe that we satisfied TA2 extremely well. We're looking for TA3, TA1, and TA2 partners in the areas listed in our slide, from facility control systems to complementary biosensor technologies to what we have today in our team, in addition to data scientists to add to TA2. So thank you for your consideration, and you can go to examer.co and learn a little bit more about our team in the team section. Thank you.
Thank you very much. Next up, we have Roger Tang from Triple Ring Technologies.
Hello, I'm Roger Tang, Chief Technical Officer of Triple Ring Technologies. Triple Ring is a science, engineering, and product development company headquartered in Silicon Valley with additional labs and offices in Boston. We partner with clients primarily in the medtech and life sciences spaces to create new technologies, launch innovative products, and start new ventures. One way to think of us is as a bridge that translates research out of academia to commercialization. In some cases, we actually create the technologies de novo. So the capabilities of our 100-person team span strategic investment, incubation, early stage R&D, product development, manufacturing, and regulatory approval, all done in close collaboration with our partners. We are ISO 13485 certified, and our experience includes many types of devices and systems. Examples of our work are on the right, starting in the upper left corner and working clockwise, include the Hound Labs THC breathalyzer. This is a handheld sample collector and detection system for detecting the psychoactive component of marijuana, THC, in breath. We have developed additional diagnostics for air and breath-based systems as well. In the upper right is a handheld device for measuring tissue health in the surgical suite using a multi-wavelength optical technique. This particular example is the second generation of the device. We took this from concept to clinical validation and FDA approval in 18 months. Below that is a system that images and positions the patient for radiotherapy in one device while the patient is sitting upright. In this case, we designed the gantry and CT scanner. The dog below is wearing a non-invasive monitor that performs long-term continuous monitoring of various physiological parameters, and it provides real-time feedback and alerts to her owner. To her left is a robot-enabled X-ray CT scanner for a horse and other large animals that provides imaging. It also includes a motion tracking system for motion correction of that imaging system.
1 minute left.
The lower left corner represents our experience with swallowable electronic devices. These smart pills have onboard electronics and capabilities that allow them, for example, to stay resident in the GI tract and to deploy when activated for potential diagnostic and therapeutic applications. Above that, on the finger, is a tiny radiation source that's used for electronic brachytherapy. Instead of using radioactive seeds, these radiation sources supply high energy X-rays for radiotherapy applications. And finally, in the middle, we have a representation of the low-cost multiplexed quantitative biomarker detection platform for a lab at home in low resource settings that we are currently developing under a BARDA contract. For the ARPA-H Breathe program, we would like to partner with anyone with early stage biosensor technology that we can help mature to the point of readiness for deployment. But we're happy to talk to anyone around any of the technical areas. Our contact information is on the Breathe teaming page. Please reach out if you have any questions, and thank you.
Great, thank you very much. Next up, we have Domitilla Del Vecchio from MIT.
Good afternoon. My name is Domitilla Del Vecchio, and I am in mechanical engineering and biological engineering at MIT. So we have developed a fieldable process for on-site detection of airborne pathogens. We have been focusing mostly on SARS-CoV-2 for reasons due to the pandemic, but the technology can be applied to detect any other virus with genetic sequences known. On the right-hand side, you see a high-level overview of our approach. So we are using a cheap electrostatic precipitator to sample the air from collecting the particles on a metal probe. Then we go through a very quick and simple RNA extraction process through eating. And then we have our proprietary RNA enrichment step that allows us to concentrate by nearly 5-fold the RNA extracted into a small volume in order to hit the limit of detection of the downstream RT-qPCR. So the current performance in our lab is a sensitivity of about 5 genome copies per liter of air, which, to give you an idea, falls in the low range of the concentrations of, for example, flu and COVID virus detected in indoor places such as long-term care facilities, hospitals, airplanes, and daycare centers. Our current timeliness is about under an hour. So what we are looking for the breath is connections and contacts in healthcare to perform our field testing. And then we are looking for entities with product development expertise and entities with engineering and manufacturing capabilities, given that at this point we have a process, but it's not yet integrated nor automated. So you can see down here, it's a reference to
1
our
minute left.
published paper. And thank you very much for your attention.
Great, thank you. Next up, we have Mark Woolham from Integrated Nanosystems Development Institute.
Hello all, my name is Mark Woolham. I work at Integrated Nanosystems Development Institute at Indiana University, Indianapolis, and I work with Dr. Mangalal Agarwal, who's the PI of the lab. And I also work with Dr. Deepak Mati. We're both research scientists. So our lab focuses on breath analysis. So we all know that canines can be used for a lot of different forensic applications like smelling drugs, smelling human cadavers, but they can also be trained to detect different medical conditions just by smelling the exhaled breath of their owner. So our lab tries to identify and detect these chemicals. For identification, the gold standard is gas chromatography mass spectrometry. So we use those assays to really try to identify specifically what are the volatile organic compound markers that the canines can be smelling. And we do that through biostatistics. But of course, this instrument is 300 pounds. So the information and the chemical identification that we get from the GCMS, we want to translate into a portable device that can sense these compounds. And some example for the GCMS stuff, the top panel in the upper right-hand corner, you can see we identified a biomarker panel of just 3 VOCs in exhaled breath by GCMS that could distinguish COVID with over 95% cross-validated accuracy. And the most interesting result came, Dr. Agarwal was pushing us to collect these samples, these COVID positive samples after they recovered, like 2 months after they recovered and they're, not experiencing any symptoms. And we ran and analyzed those and all the VOCs come back to healthy baseline levels, which is really interesting. So as far as sensors go, which is really the interesting part and the focus of ARPA-H, we have 2 different arms. We're developing our own breath-based biosensors, and we identified many different sensing layers and fabricated many different devices that can sense a handful of VOCs that we've identified in our GCMS studies. But then we also we partner with industry and take off-the-shelf components like these large sensor arrays, and we test them as well with real breath, simulated breath as well. And both of these platforms we have tested and validated, and we can really tune the machine learning algorithms and train them and validate them to detect the already identified VOCs of bacteria, viral infection, and fungi. The overarching vision is that we're going to passively monitor VOCs emitted from bacteria and fungi because they actually emit VOCs as they grow, and the VOCs are pretty well understood. However, viral infections do not emit VOCs. So here we're going for breath-based biomarkers in the host. And we can do that passively by setting up a sensor in a room to monitor a group, or we can even do that on an individual basis using a reusable breathalyzer.
We are out of time.
Perfect. That's all I have. And thank you so much for your time.
Thank you. Next up, we have Lou Radonovich from the National Institute for Occupational Safety and Health, or NIOSH.
Good morning or good afternoon, wherever you may be. Thanks so much to ARPA-H for hosting this important program. I'm representing the Respiratory Health Division at the National Institute for Occupational Safety and Health, or NIOSH, at the Centers for Disease Control and Prevention, or CDC. Our specific area of interest in this solicitation is Technical Area 3, or leveraging protective and responsive building interventions to reduce bioaerosol exposure risk at optimal costs. So at the Respiratory Health Division at NIOSH and CDC, we do multidisciplinary research to identify work-related respiratory hazards. We assess workplace exposures. We characterize health risks, and we develop and disseminate effective interventions. We're primarily interested in this solicitation as it pertains to germicidal ultraviolet, or GUV, as an intervention to reduce indoor transmission of respiratory infections in workplaces and other indoor settings. Since 1990, we at CDC and NIOSH have recommended GUV air treatment to augment building ventilation systems to reduce germs in the air. In 2009, NIOSH published Environmental Control for Tuberculosis, Basic Upper Room Ultraviolet Germicidal Irradiation Guidelines for Healthcare Settings, as pictured here. This report became the design standard for safe, effective GUV systems in the United States and many other parts of the world. We, in a collaboration with our NIOSH and CDC colleagues, have published numerous other documents pertaining to GUV and ventilation. So to complement our expertise in GUV, ventilation, human health risk assessment, translational science, and clinical search, we seek partners with expertise in indoor air biosensors, or TA1, and respiratory risk assessment software, TA2. My contact information is here by email, and I'd also direct you to the CDC website for additional information that we've disseminated. Thanks for the opportunity.
Thank you very much. Next up, we have Gregory Vasquez from Detect Ion.
Hello. Detect Ion is a startup that was founded in 2021, Tampa, Florida. Our mission is next generation CBRN sensors. Our vision is democratizing mass spectrometry. For Breathe, we envision leveraging VOCs for infection detection. We're interested in partnering within TA1, aiding mainstream biotech sensors. Detection is real. Breath detection is real. The left panel shows results of the paper testing VOCs for COVID-19 detection. Their thermal desorption GC mass spec data show that the VOC profile was able to detect individuals with infected COVID-19 with variant level identification. There were also no false positives per PCR-based confirmation. Interestingly, it shows better ROC curves for the individual variants than the combined total. This provides proof of concept for detection of respiratory infection using VOCs. Detect Ion developed a chip-scale mass spec as part of a pre-concentrator thermal desorption GC-MS detector platform. It has low size, about 10 liters, it's a low weight and low power consumption, all of which is considered SWAP. We've achieved mass spec on demand, going from passive to active within 30 seconds. We can achieve parts per trillion detection within a few minutes. Development was funded by DARPA, ARPA, and DTRA for CBRN sensing. In a government field test, it detected and identified all test chemicals, including the true unknown. It's been ruggedized and successfully deployed on an aircraft, an Air Force C-130, for real-time detection of methyl salicylate, which was the first time for a mass spec, and deployed in subways for DHS. We're aggressively developing point-of-care diagnostics and currently funded by the Gates Foundation for pilot development of malaria and TB-specific detection. We have a working, proven detection platform. Our breathe approach is early detection. Since our detection platform has minimal consumables, it can be used as a primary VOC detector to establish presence of infection within a room or building sector. This can trigger TA1 pathogen-specific detectors with reagent and processing time limitations for direct identification of pathogens. Simultaneously, it can trigger TA2 to isolate HVAC in those areas and begin TA3 remediation. The same device can be used to sample individuals in the affected building areas for infection for isolation or treatment. Strategic goals are, we're seeking partners across all TAs, we're open to priming or subbing, and we're open to cost share scenarios. Thank you.
Thank you very much. Next up, we have Jeff Bounds from Kitware.
Hi, I'm Jeff Baumus, the Director of Data and Analytics at Kitware. For the BREATHE program to be successful, the Indoor Air Quality Index has to be not only accurate, but must obviously also be usable. So BREATHE needs effective software for organizing the sensor and other data and presenting the information in comprehensible metrics and visuals. That's where Kitware comes in as an expert in data and visualization software to mediate between the complexity of the sensor and other data coming in from TA1 and the people on the other side, TA3, who want to make sense of that data. Kitware is a software company with extensive experience working on custom solutions alongside broader research teams. The data and analytics team in particular is fluent in designing and developing scientific data systems. We have a strong focus in data fusion and organization, in addition to building custom scalable data visualizations. We have extensive experience in biomedical and spatial data, which will be relevant here on the BREATHE program. For example, we're currently working on a web portal for a diverse set of microbiome data. We're also building a layered urban planning application with custom spatial visualizations, which integrate with climate simulations and other urban data sources, such as population density. We expect BREATHE to have similar models and data sources to deal with. We've been part of several DARPA programs in the past, including programs for large data visualization, custom search engines, and novel ML interfaces. We have visualization experts and UI UX designers on staff, and Kitware has a strong breadth of other technical expertise, including AI, ML, scientific computing, and medical applications. For the BREATHE program, we're a good fit for a more maybe basic research heavy team that needs expertise in building interactive visualizations and robust data system software. We build the interfaces both for domain scientists, which are more TA1 facing, and system operators in buildings, which is TA3 facing. We would expect to need a partner in TA2 for the specialized analytics and modeling involved. So the first few headings under TA2 and RFP, but we'd handle a lot of the rest, which would be focused on integrating those models with scientific dashboards, visualization, data management, data infrastructure, data fusion from other sources, integrated analytics and models, and doing usability studies. Please feel free to reach out if you want to talk about teaming. My contact information will be up on the teaming profiles table online if it's not there already, or you can email kitware@kitware.com. Thanks.
Great, thank you very much. Next up, we have Danny Mosh from Green Vision Systems.
Thank you for letting us doing this lighting. My name is Danny. I am the founder and the CEO of the company. Just want here to brief about what we have been done over the year. Developed a system that can sense air from indoor air and provide a different type of information. It's a system developed for online automatic air sampling. And after the system have done the cycle of air sampling and provides a result within 5 to 10 minutes. So it's a near real-time system, can work automatically in a different type of scenario and environmental condition. As can see here, as the system can detect different type of bioaerosols, such as bacteria, viruses, fungus, and specific allergens. And the system have also anomaly detection capability, which can add a specific value for better controlling and reducing biotrace in a specific environment. The high capability of the system that enabling us to generate result as indicate here, based on the ongoing adaptive databases, which are learning the background of the specific environment and actually focusing on the targets that we are looking for. As can see in the example here, you have two major examples. One is we have been sensing in different patient room of COVID SARS-2, as those different number of room have different, let's say, viruses load. So the system have been sensed in reference, as you can see here, the small green here, and also in the different viruses loads, and the correlation which have been then done with RT-PCR, and you can see it here. We are generally in a TR level 8, and we're looking for a strategic partner to take what we have been developed to implement our solution in TA1. Thank you.
Thank you very much. Next up, we have Louis Goudron from University of Hertfordshire.
Good afternoon. Thanks a lot for having me with you today. My name is Louis Goudron, and I'm really happy for this opportunity to present the portfolio of our Center for Research in Biodetection Technologies. Hopefully, I will highlight how we can contribute to breadth and specifically on delivering on TA1. Our center is part of the University of Hertfordshire, who is one of the top UK universities working on bioaerosol detection research. We have established a 15-year track record of delivering solutions for the next generation biodetectors for application ranging from plant pathogen monitoring to human protection from biological warfare agents, often working in close collaboration with other universities and UK government agencies. Recently, we have earned £13.5 million of funding for the University of Hertfordshire to create and lead the future biodetection technology hub, gathering other top UK universities at Cranfield, Manchester, and Leeds in order to develop UK research in biodetection of airborne pathogens. Our team at Hertfordshire is highly multidisciplinary, with expert scientists working hand-to-hand with electronics and mechanical engineers to design, manufacture, and test what we refer to as work turnkey prototypes. Broadly speaking, our systems aim at responding to airborne detection challenges in all three stages of sample collection, sample recovery, and detection and analysis. For bioaerosol collection, depending on the size range, size fraction, and the use case, we have developed a range of collectors using different collection mechanisms like electrostatic precipitation, cyclonic separation, and filter collection. To work with those collectors, we have developed a variety of sample recovery systems like foam filter extraction system or digital microfluidic system that allow to control the movement of single droplets. Digital microfluidics is used in conjunction with electrostatic precipitation to elute aerosol material collected on the plate into a super concentrated small volume droplet. When it comes to detection and sample analysis, digital microfluidic is also quite useful as it offers full lab-on-a-chip capability with tiny volumes, which enable fully automated detection platform like our immunoassay platform shown here. An important aspect of our recent work is aimed at integrating the several stages of aerosol detection into one system. On the left, we have used automated mechanical translation of the collection plate to merge electrostatic precipitation with digital microfluidics for integrated bioaerosol collection and sample recovery. On the right, the dry sample detection unit allows us to take the electrostatically collected plates and use it directly as an input sample for digital microfluidic DNA amplification and detection platform, including a fully integrated droplet elution steps. Obviously, the smaller the system, the more challenging the integration. With bigger systems like the 7-day sports century, integrating cyclonic separation, the 3 stages of biodetection can be integrated to perform pseudo-continuous monitoring of plant pathogen with flame detection. This brings me to the end of my presentation. I hope I was able to convey how a set of skills can be useful to address Tier 1 challenges, and we're looking for collaborating to complement partners for Tier 2 and Tier 3 and other specific aspects of Tier 1. My email address is on screen. Please get in touch if you wish to discuss further. Many thanks for your time.
Great, thank you very much. Last up, we have Yao Yang from Texas A&M University.
Hi, this is Jiao Yang. I'm from mechanical engineering in Texas A&M University. I'm part of the O'Neill group. We have 3 themes of expertise in our group that's closely related to the BREEZE program. The theme 1 is a full stack energy, building energy, and the indoor air quality performance assessment. We have a long history of working closely with the building stakeholders in the building sector, and we have a lot of experience in and also impact on how buildings are controlled, the ventilations are controlled, especially for the indoor air quality. So 2 examples. One is for the demand controlled ventilation, where we use the CO2 as the proxy for the occupancy and also contaminants. We dynamically adaptively control the ventilation for those purposes, and it's the simulation is also implemented in the test in the demonstration in a commercial building. Second example is the smart mechanical ventilation to mitigate the risk of infectious disease. This is a purely simulation study, and what we have found is also incorporated in the ASHRAE standard 241 control of infectious aerosols. The theme 2 is controlled laboratory testing for building energy performance and indoor air quality. So we have a very unique lab home test bed built in Texas A&M. These 2 homes are identical, fully instrumented with sensors and also emulators for occupant activities. So what's unique about it is we can reconfigure the ventilation scheme and the control, and also we can reconfigure even the floor plan to study both a typical residential home or a larger open space that's found in like commercial buildings like schools. The theme 3 is a field demonstration of building energy and the control systems. So we have a lot of experience in quick iteration and field demonstration of advanced sensors and controls to connect them with all types of building automation systems and use the sensor information to drive the control decisions for the ventilation systems. So this is a particular ARPA-H funded project. We work with our sensor team, and we put several iterations of prototype sensors in the buildings for occupancy-centric controls for both energy and thermal comfort. So we deploy them in apartment buildings, residential buildings, and offices. That demonstrates that we have a capability to work with the different novel sensors. We also can have data-driven decision-making based on the sensor data and also analytics. So our information, our contact is on the screen, and feel free to reach out. Thank you for your time.
Great, thank you very much. And thank you to all of our lightning talk presenters, and thank you to everybody for joining today's webinar. As a reminder, we've recorded today's webinar, and we will post the slides and the recordings on the BREEZE program page once those are available. Please continue to check the BREEZE website listed on this slide for program updates, including additional FAQ or Q&A updates, and for when the final solicitation will be posted within the next coming weeks. If you are interested in submitting a solution summary or proposal and would like to find potential teammates, you can also visit our teaming page. The solution summaries and proposals should address all 3 technical areas, so we encourage you to reach out to potential teammates if needed. This concludes the webinar, and I hope you have a great rest of your afternoon.